Well, hello and welcome back to our second video about carbohydrates. In this video, we'll be focusing on disaccharides and polysaccharides. As a reminder, when we introduced monosaccharides, we described how carbohydrates in general can be classified or categorized into one of three categories. Those three categories are monosaccharides, disaccharides, and polysaccharides. In the first video, we talked about monosaccharides. In this video, we'll be talking about disaccharides and polysaccharides. And in particular, here's what we will be doing. We'll be looking at reactions of disaccharides, oxidation, hydrolysis, and condensation, and also learning with structures of disaccharides and polysaccharides how to identify glycosidic bonds. So to start, I'm going to ask you to flip back in your workbook to page 161. That's where we ended the previous video. So if I look at the bottom of page 161, you'll notice that there's a monosaccharide drawn here. In the previous video, you learned how to identify this as a monosaccharide. You learned how this is a aldohexose. I briefly, in that video, described how in solution, when monosaccharides are dissolved in solution, a rearrangement reaction happens where the acyclic form of that monosaccharide, this is acyclic, it's not a ring, it rearranges to form a ring, a cyclic form. This oxygen here bonds to this carbon here. So there's the oxygen, there's the carbon, a new bond forms. And then the double bond that previously was part of the aldehyde goes away, and this becomes an alcohol. Notice this is no longer an aldose. It's not an aldehyde or a ketone. This rearrangement, by the way, is what allows all monosaccharides to become oxidized, whether it was an aldose or a ketose. All monosaccharides can get oxidized because all monosaccharides exist in equilibrium between the acyclic form and the cyclic form. Again, you cannot see from this form that it used to be aldehyde or if it had been a ketone. Ketones do a similar equilibrium. You don't need to be able to describe the details of this rearrangement, but what I do want you to recognize is that monosaccharides exist in both the acyclic form and the cyclic form. It's the cyclic form that will be helpful for us in being able to show reactions of the monosaccharide. So again, this is a monosaccharide, this is also a monosaccharide. Now let's return to where we started. We were describing on page 166, starting on page 166, disaccharides and polysaccharides. We want to look at oxidation, hydrolysis, and condensation reactions of these. So I'm going to flip the page to 167. Have you please join me on page 167. If we were in class together, I would point at this and I would say, what is this? Is this mono, di, or poly? And I would want you to recognize that this is a monosaccharide. It's a single ring. Mono means one, and this is one ring. It does not refer to one carbon. There's 6 carbons. It refers to how many rings. This is one ring. So that's a monosaccharide. This is also a monosaccharide. The reaction that I'm showing you right now is going to help us see how do disaccharides get formed. So a monosaccharide plus a monosaccharide will undergo a condensation reaction. In the direction I'm writing it right now, this is a condensation reaction so that a monosaccharide plus another monosaccharide can combine. Condensation is a type of reaction that you saw for the first time in the formation of esters, the ester reaction, ester formation reaction. Here's another example of condensation. Sometimes what I like to point out is this. The official name of the reaction is condensation. It starts with the letter CO. Let that remind you that condensation means to combine. It means when you're combining two different structures to form one larger structure. Specifically, the way that that condensation happens, The way that that condensation happens is box out water. Draw a box between OH of one of the functional groups and H of the other. The OH from this monosaccharide and then the H from this monosaccharide. I'll point out I could have chosen this H with this OH. I'll get the same product either way. So it doesn't matter if you chose OH with H or H with OH. You'll get the same thing. What you cannot do though is box out OH and then also OH. That's too many O's, because as you know, what I just boxed out becomes the second product, water. That HHO is this HHO. So we've boxed out water. You've seen that type of condensation reaction before when we formed esters. In this case, though, this is not an ester. It's no longer a monosaccharide. It's now a double, right? There's two rings. This is a disaccharide. So what we did is we combined a monosaccharide with another monosaccharide to form a disaccharide. That reaction type is called a condensation reaction type. And as you see, we formed a new bond. When we were making esters, we called that new bond an ester bond, but this is not an ester. What this is a disaccharide, which is a carbohydrate. In carbohydrates, the new bond is called a glycosidic bond. So that is a glycosidic bond. That's the new bond that gets formed when we formed a disaccharide. Once you do that, now we have a disaccharide, and you ought to be able to identify by structure different carbohydrates as monosaccharide, disaccharide, and so on. Here's the most common disaccharide that we encounter, sucrose. Sucrose is otherwise known as table sugar. In other words, what you might bake cookies with or add to coffee. That's table sugar, sucrose. This is a disaccharide. I can see that because I can see that it has two rings. If I were to ask you right now to identify the glycosidic bond in this structure, it's right here. That's the glycosidic bond right there. Same with the two structures below. This is also a disaccharide. And the one below it, maltose, is also a disaccharide. I can see the glycosidic bond for each of these. Here's the glycosidic bond for maltose, and here's the glycosidic bond for lactose. What I've highlighted in each of those cases is the bond, which is carbon, oxygen, carbon, or carbon, oxygen, carbon, carbon, oxygen, carbon, and so on. That's the glycosidic bond. Notice the monosaccharide does not have any glycosidic bonds because they have not yet connected with another monosaccharide. No glycosidic bond present in a monosaccharide. There's not a glycosidic bond in a monosaccharide. There's one glycosidic bond in a disaccharide. So I'm going to, at the top of this page, I'm going to keep track of the reactions that, that disaccharides can do. We're going to look at each of them right now. We have not yet done any. The reaction I did down below was reactions of monosaccharides. So in fact, if I do this same thing, reactions of monosaccharides, this is from the first video. We first saw that they can do oxidation. We saw that in the first video. And then what we just saw right here is that monosaccharides can undergo condensation. I want to show you the most common reaction, or a common reaction, of disaccharides. And there's a total of 3 that we will look at. I'm going to list them here. We'll talk about them probably in a different order, but we will talk about all 3 right now. Oxidation, condensation and hydrolysis. Those are the same that I listed on the objectives of the very beginning of this video. So we already looked at oxidation in monosaccharides, and we just now looked at condensation reaction in monosaccharides. Let's do the same thing for disaccharides. Oxidation, condensation, and then hydrolysis. I'm going to start with hydrolysis, and you'll see why it makes sense to start with that one. It is just the reverse of condensation. Again, we saw that with esters. When we form esters, that was a condensation. And then when we pull them back apart, that's hydrolysis. So hydrolysis is just the opposite, the reverse of condensation. If we were in class together, it would be easier to show you this, but I'll still try with this video. Sometimes what I say to kind of help keep things straight in your head, I say, hey, everybody, look at the ending letters of that word, hydrolysis. Hydrolysis, sometimes I say, let that remind you of the word scissors. Hydrolysis is like scissors. Scissors are not used to combine something. Scissors are used to cut something apart, break it apart. That's exactly what hydrolysis is. It's not combining, that's condensation. Hydrolysis is like scissors, chopping something up. And sure enough, disaccharide, if you use scissors, if you hydrolyze, do hydrolysis on that glycosidic bond, now instead of a disaccharide, you just pull it apart. And when you pull this apart, you go back to having the two separate monosaccharides. So disaccharide can do hydrolysis, which is just the reverse of condensation. You should know that. That definitely is something I'd like you to know, that disaccharides, yes, can do hydrolysis. You won't have to draw out the structure. In other words, if I were to say complete this reaction, if I tell you I'm going to do hydrolysis on this, I'm going to attempt hydrolysis on this, and I ask you to complete the reaction, here's what you would need to be able, how you would need to be able to do that. First, you need to answer yes or no. Can this do hydrolysis? It's a disaccharide, so yes, we can do scissors on it. We can break that glycosidic bond. We can pull it apart. What happens when you break apart sucrose? You could answer this one of two ways. You could answer it to say, well, that was a disaccharide, so I know that I will end up with a mono plus a mono. That's true, and that's pretty good. I think you can do a little bit better than that. In fact, I'm asking you to be able to do a little bit better than that. Specifically, I want you to know which two sugars do you get when you hydrolyze sucrose. Sucrose is a disaccharide. Which two monos specifically do you get? And the answer is this: sucrose is formed from a glucose plus a fructose. When you do Lab 9, sugars and starches, you'll work with that in quite a bit more detail. But in general, I want you to know that when you combine a glucose and a fructose, you get a sucrose, which means when you break apart a sucrose, what you get back is glucose and fructose. That's hydrolysis. Pay close attention to this reaction right above. Water is a product with condensation, which means when you run everything in reverse, water will be a reactant. So there will always be water here as a reactant if you're doing hydrolysis. That's so that we can put H and OH back when we pull this apart. We're not going to focus on that or worry about it or pay much attention to it, but to have a complete reaction, we do need to list it. Water is a reactant. Disaccharide undergoes hydrolysis to get a mono and a mono. If the disaccharide happened to be sucrose, then the products will be glucose and fructose. If the disaccharide happened to be lactose, that's the second of the three that I listed at the beginning of this video for you, I would like you to know which monosaccharides do you get when you hydrolyze lactose. One of them is glucose. Glucose is kind of a famous, common one. The other one, it's a little bit given away by this name, lactose, it's galactose. Galactose is one of the three monosaccharides that I want you to kind of become a little familiar with just because of its role in this reaction. Glucose is a monosaccharide. Galactose is also a monosaccharide. When you break apart lactose, you get the mono plus a mono. If you wanted to build a lactose molecule or make a lactose molecule, you would do it by combining galactose with glucose. Combining is the other direction, which would be condensation. Last but not least is maltose. You can see that it's a disaccharide, which means you know that it, yes, can undergo hydrolysis. And when it undergoes hydrolysis, you will get a monosaccharide plus another monosaccharide. The two specific monosaccharides you get when you do that are glucose and another glucose. So glucose, as you see, is common. It's not true that every single disaccharide will produce a glucose when it undergoes hydrolysis. That's not universally true, but for these three particular disaccharides, it is true. So I'm going to slide the paper up a little bit. What we've just looked at is hydrolysis reactions of disaccharides. You will not have to, most commonly, you will not be needing to draw all of these structures, but you very definitely need to interpret them and make sense of understanding them, as well as being able to answer with names of compounds like we've just done. So that is hydrolysis. I would like you to consider, though, what about condensation? Could this keep going to make another bigger molecule? In other words, could we make a trisaccharide or even bigger? Yes, the answer is yes. And let's look at that on the, I think it's the next page. Yeah, sure, right here. The next page, page 168, shows polysaccharides. A polysaccharide is produced by combining many monosaccharides. So in other words, a disaccharide plus a mono would give you a tri. And if you do, I'll kind of just make up a little bit of a number here. If you do thousands of monosaccharides combined, that's how you get a poly. We obviously are not going to draw it out. When I say the word many, I mean thousands, hundreds of thousands, lots. We can't draw that out. So what I've drawn here is a little snippet. This means that little end there means it keeps going. This means it keeps going in the other direction. We've just kind of zoomed in on just a little bit of the molecule. There's a ring, there's a ring, there's a ring, there's a ring. This is a polysaccharide. Another polysaccharide. See if you can find glycosidic bonds in this structure. Pause the video and see if you can highlight or circle the glycosidic bonds present. And here they are. This is a glycosidic bond, glycosidic bond, glycosidic bond, glycosidic bond, and so on. We noticed a few moments ago that a monosaccharide has 0 glycosidic bonds present. A disaccharide has exactly 1 glycosidic bond. Polysaccharides have a lot of glycosidic bonds. The polysaccharides that I listed at the beginning of this video, starch, glycogen, and cellulose, I'm listing them on these pages. You do not need to be able to identify them by structure. I just put the structures here to show you. But what you do need to know is starch is a polysaccharide. And if we flip to the next page, I show glycogen. I want you to know by name that that is a polysaccharide. And I want you to know by name that cellulose is a polysaccharide. I also want you to be able to do this. If I show you a structure like this, or like that, or the ones on the previous page, you should be able to say, yep, that's a polysaccharide. You don't need to say which specific polysaccharide, but yes, be able to identify it as a polysaccharide. And again, also be able to identify the glycosidic bonds. There's a glycosidic bond, there's a glycosidic bond, there's a glycosidic bond, glycosidic bond, glycosidic bond, glycosidic bond. One of the things we did have to cut out of this topic when we lost our couple of weeks due to this COVID-19 is we cut some topics out. One of those topics was identifying glycosidic bonds as alpha or beta. You don't need to know that. I'm going to cross that out. We're not talking about that. So polysaccharides are huge. If I come back to the page that talked about reactions of mono and disaccharides, I did not list hydrolysis is like scissors. Can you chop it up? Can you break those glycosidic bonds? The answer is yes. That's in fact exactly what happens when you eat starch. The first thing your body does, in fact, it actually starts in your mouth. Those glycosidic bonds start breaking to make that giant polysaccharide smaller and smaller and smaller and smaller. When it gets small enough, when it gets down, for example, even to a disaccharide stage, I'm going to flip back to the disaccharide page, page 169. Here's page 169. You might eat a disaccharide directly, for example, eating sucrose or lactose or maltose, or you might have those fragments formed from chopping up a polysaccharide that you ate. But if you have disaccharides in your body, we saw earlier that they can do hydrolysis. We saw that at the beginning of the video, but you can also add on to a disaccharide. So disaccharides can do condensation by boxing out water to make a big molecule. So how would you ever make a polysaccharide in your body? By keeping on building, like adding more and more and more and more monosaccharide rings, even starting at a di. So hydrolysis happens in your body. We showed how that happens. Condensation of disaccharides can also happen in your body to make something even bigger. The final reaction, I didn't draw that. As I said in the previous page, with polysaccharides, we won't draw how you make a polysaccharide because it's too big, but it's the same kind of reaction. It's a condensation, box out water and form a glycosidic bond. So that's exactly what this is, condensation. The last reaction I do want us to talk about, because it relates to how you get the energy from the food you eat, is oxidation. It's not a new reaction type. It's what we've been learning about for quite a few weeks, but I'd like to show it to you with disaccharides. We saw it in the first video with monosaccharides. We learned that all monosaccharides can undergo condensation. What about can undergo oxidation, sorry. All monosaccharides can undergo oxidation. What about disaccharides? Can all disaccharides undergo oxidation? I'm going to highlight the specific ones we have on this sheet and then generalize it to all disaccharides. This disaccharide structure, specifically sucrose, if you were to pause the video and I ask you to stare carefully at this structure and how it looks different from the other two, what you would notice is that it's right here. This structure here, look at the right-hand side of the molecule. This carbon right here that I'll put a little blue dot on, this carbon that I'll put a little blue dot on, and this carbon that I put a little blue dot on. If you look at this blue dot, it's bonded to an O and another O. This is bonded to an O and to another O. This one is bonded to an O, but not to another O. That O is way over here. You don't need to know that that's the reason, but I'm just sharing with you that that's the reason. You don't need to know that why that I just said, but you definitely need to know the what. And here's the what. Sucrose cannot get oxidized. Again, if you're curious, the why has to do with that bonding structure. It cannot get oxidized. All other structures have the bonding arrangement of that carbon is bonded to O and to the other O. Lactose can get oxidized. And this is called a hemiacetal. That's not a functional group that you need to know for this course, but the fact that these two sugars have that arrangement and sucrose does not is the key thing here. Sucrose cannot get oxidized. All the monosaccharides can, and actually all the other disaccharides can. Lactose can, maltose can, any other disaccharides are a yes, they can, but sucrose cannot. And what we learned in the first video is what that means is sucrose is not a reducing sugar. I'm making a big point of that because that has impact, first of all, in the lab that you're doing on this topic, but also what happens in your body. Lactose is a reducing sugar, and maltose is a reducing sugar. Anything that can get oxidized is a reducing sugar. Sucrose is the only one. Of all the sugars, sucrose is the only one that is not a reducing sugar. It's the only sugar that cannot get oxidized. I want you to pretend now for a minute that you just ate something sweet and it's in your mouth. You want to get energy from it. The way that we get energy from our food is to oxidize it, but you can't oxidize sucrose. It doesn't get oxidized. So what can sucrose do? I'm going to pause the video and have you think about this. When you eat sucrose, what can you do to it? Yeah, you're right. You cannot oxidize it, but it can do hydrolysis, right? You can hydrolyze sucrose. It happens in your mouth and then later on in your stomach. It gets hydrolyzed, turns into glucose and fructose, and then these can get oxidized. So you can get energy from it. The oxidation is what gives us the energy. So if we cannot oxidize sucrose directly, no problem. We pull it apart to get the monos, and then we can oxidize those. Likewise, if you eat something like bread or pasta or rice, tortillas, eat some sort of starchy food, potatoes, those molecules are gigantic, those polysaccharides. They're way too huge to get oxidized directly. So what happens to those? Let's pull the page to a polysaccharide. What happens to the gigantic polysaccharides that you eat? Well, hydrolysis. It starts getting all scissored up, do hydrolysis on all those glycosidic bonds to make them much smaller. And once they are much, much, much smaller, either to the disaccharide or the monosaccharide stage, then they can get oxidized. The only sugar that cannot get oxidized is sucrose. So let me do this. Let me pull back up our original paper, page 166. What we wanted to do is identify structures for disaccharides and polysaccharides. We did that. We identified glycosidic bonds in disaccharides and polysaccharides. And then we looked at oxidation reactions, hydrolysis reactions, and condensation reactions for disaccharides. The only reaction of polysaccharides that comes up is hydrolysis. Chop it up, use scissors to make it smaller. I haven't even listed that on here, but I did put it on those notes. And last but not least, before I end this video, I want to highlight something to make sure you get it. These are considered sugars. The disaccharides and the monosaccharides are considered sugars. They're sweet on your tongue. Those are sugars, and notice they're ending os, 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 os. Sugars end in os. Polysaccharides are not considered sugars. They generally, well, they don't taste sweet. And they don't end in os except for this random one. Cellulose, even though it ends in os, it's not a sugar. Too bad that we don't get to be in charge of that. We'd probably pick a different name for it so it doesn't look confusing. Even though it ends in os, it's not a sugar, it's a polysaccharide. It's also just fun fact, this is not the same thing as cellulite, stuff that might be on your body that you don't like. It's totally different. This is cellulose. This is used for plant carbohydrate structure. This is like, think celery and tree bark. This is used for structure in plants. This is plant carbohydrate storage. So let's think of a potato. All the starchy stuff, the white part of the potato is starch. The skin of the potato would be made from cellulose, its structure. Or a corn kernel, if you eat corn, The outside hull is the structure that would be made from cellulose. The juicy stuff on the inside is made from starch. So storage versus structure. Your body, like human bodies, cannot digest this. I won't get too graphic for you, but go eat some corn or eat some skin on potatoes and then just take a look later whether or not that actually digested. It's because you cannot digest cellulose. You can digest starch. What's glycogen? That's how animals, including us, store carbohydrate. You need to have access at all times to glucose, but you don't want to have that much actual glucose in your blood. So we store glycogen in our liver to have access to glucose. Glycogen is big, so as you know, if you want to get that smaller, you do hydrolysis. And that's what we want to say with disaccharides and polysaccharides. I'll end the video now, and there's plenty of practice problems for you in the workbook. Thanks for joining me. Bye-bye.